Just to review this, this just shows you the different types of antigen-presenting cells that you can have. So we've got the dendritic cells, and I just wanted you to take note of what types of antigens do they deal with, and then where do we find them. So these dendritic cells are going to be all throughout the body. They're going to be in your tissues, and your blood, and your lymphatic system, etc. Macrophages, notice here, connective tissue and body cavities. Macrophages, why we talk about these most is because this is what we're going to find in our lungs and things like that. And they're there to rid us of the pathogens that get through that first barrier of defense. And then finally, we've got the B cells, and these are going to be moving through the blood and the lymphoid tissue. And these are originally produced in the bone marrow, but these are going to be where we initiate basically our specific defense. So let's talk about the types of lymphocytes that we're going to be looking at. The first ones, and probably the most important, so when you guys go back to the immunity for SL, you'll see that it starts talking about HIV. So, okay, I'm gonna go ahead and pause this. Making a screencast. Message you when I am done. Your messages are showing up. Okay, so CD4 helper cells, helper T cells, are arguably one of the most important cells in your immune system. And the reason for this is because they are basically the event coordinators. So if you think about like a wedding planner, right? What would happen to a wedding if all of a sudden the wedding planner was taken out somehow? And so think about all of the things that they do. They basically cause everything to happen. And that's what the purpose of this CD4 T helper cell is. So basically, it starts out, they're produced in the thymus. So this is part of your lymphatic system. And you'll find them in lymph nodes and other lymphatic tissues and organs. And the naive T cells, so they both, B cells and T cells, start out as naive T cells. And basically think of them kind of like undifferentiated stem cells in a way, because they both retain the ability to divide and proliferate. They then will differentiate into different types of cells. And they currently are open to many, many, many different types of antigens initially, due to the fact that they have so many different proteins that are in their membranes waiting to coordinate with a specific antigen due to alternative mRNA splicing that we talked about back when we were doing post-transcriptional modifications. So it's good to go back and kind of take a check on that and make sure that you kind of understand what's going on there. But essentially think of this as you've got a T4 helper cells. Now, if for each, there's tens of thousands and thousands of different receptors that are going to be there for you to be able to meet up with a specific antigen that exists out in the environment, right? And they didn't have these antigens present beforehand. This is all done simply through that random splicing. So these different receptors, there's only going to be a few cells that are going to have a specific receptor if you've never been exposed to that particular antigen prior to its primary exposure. So think about it kind of like that. So that's why it's kind of slow right off the bat for your body to develop an immune response against an antigen that you've never been exposed to before. So the helper T cells are what's about to come in. Here's a helper T cell. And helper T cells, this is a naive helper T cell. It is inactivated right now. And you'll notice that it's got these receptors on the outside. And helper T cells, due to the alternative mRNA splicing, it is going to have receptors that match up to a bunch of different antigens on its surface. And this is in order to find it. Not all of them are going to have the right receptor for this. And so there's only going to be a few, if this has never been introduced to the system before, there will only be a few helper T cells that will have the right receptor that will match up to this particular antigen. And so what's going to happen is this receptor is going to receive this antigen and form a complex between the APC and the T4 helper T CD4 plus helper cell. When they bind like this, it's going to trigger these proteins to be released from the antigen presenting cell called cytokines. And these cytokines are going to be released, and these are going to help to activate the helper T cell. So you can see there's the cytokines releasing, and it's going to then cause this helper T cell to proliferate. So it is now going to make lots of copies of itself, but it's also going to produce more of these chemicals that are going to activate. When there is only one or two antigens that are presented, and this doesn't happen by a bunch of different macrophages, then eventually these T cells are going to just destroy themselves if it doesn't reach a threshold. So that's partly why it keeps sending out these signals. These signals are not for other cells, it's to activate itself. And it needs to be a certain threshold for this to reach before it will actually proliferate and form all of these different activated T helper cells. So here it is proliferating. And now that we've got these are going to go into the effector phase, which is when we now need to activate the cells that are going to be responsible for the antibodies. So once we have our activated T cells, it's time for them to start coordinating events. And the biggest thing that they can do, their biggest role, is activating cells that are like the cytotoxic killer cells that are going to kill, for example, cancerous cells or cells that have been infected by viruses, but the viruses are still inside the cells. They also are responsible for the activation of the B cells. And the B cells then are going to go through a cascade of reactions that's going to cause them to produce antibodies, which are really the key to our human immune response. So B cells are produced in the bone marrow. They are phagocytic. They have receptors with antigens that form a complex with the T cell to become activated, and you'll see this in the video in a second. And once activated, they divide to produce memory B cells, displaying a specific antigen, and plasma cells, which differentiate produce massive amounts of antibodies. They are then released in the blood and circulate, and then we'll talk about what antibodies do a little bit later. And as we said earlier, think about which organelles you would expect to see in high number in these cells, right? So what is the purpose behind that? So think structure, function, structure, function. So one thing that you need to know is, again, this is another kind of checks and balances thing. We want to make sure that there is a significant concentration of this antigen in the body before initiating a very energy dense, requiring response. So in addition to this, the B cells need to first be activated. And again, B cells show the similar type of, they have lots of different types of antigen receptors, or these look just like the antibodies actually, on the outside of their surface. And again, they're going to become activated once they attach to a specific antigen. And what that's going to cause the B cell to do is to differentiate. And for both of them, when they differentiate, they end up only presenting one type of receptor. So even though they may have started with tons of different receptors, they're only going to have one type of receptor once they are activated. And that receptor is going to be specific to this particular antigen. IgM stands for immunoglobulin, and that's also what we call an antibody, right? And notice that they also have these MHC class proteins. Because it is a cell that's phagocytic, it's going to do the same exact thing that we saw the macrophage do. So there's the phagosome, the phagolysosome, digest the bacteria. The antibody with the specific variable region that's going to be specific to that antigen is then going to be presented by the MHC class 2 protein and displayed. And once it does this, it can now interact with the receptors that are on the activated T helper cells. So the activated T helper cell then is going to attach, and once it does, it is going to release cytokines to activate the B cell. And the B cell then is going to differentiate and divide in mass amounts, lots of mitosis happening here, to produce these cells called plasma cells, as well as memory B cells. These are now going to be differentiated so that they can produce loads of antibodies that are specific to that one antigen. Now, if you think about this, inside of there, they're going to be, first of all, low gene expression, because the purpose of this plasma cell is literally just to live to produce one specific antibody. So the gene expression is going to be limited. In addition to that, because we are secreting proteins, then all of the organelles that we associate with protein secretion, so the rough endoplasmic reticulum, Golgi apparatus, etc., are going to be an incredibly high number in these particular cells. And that all occurs once differentiation happens. I really apologize for those notifications, but I'm not reading this. So the plasma cells now are going to release antibodies, and these are going to go out into the blood, and they are going to then go into our next phase, which is, what do antibodies actually do? How do they help us control pathogens in the body?